also record the location.
What is this app?
It's called
iNaturalist?
let me show you. So these are all my pictures. I have a lot of pictures. It's called iNaturalist.
iNaturalist. Okay.
So
How it works?
so if I, for example, I see
For
this,
this
this
sec.
bag
Yeah.
on the ground. Oops.
Right.
Yeah, even if it's like that.
Right.
I can take a picture.
Right.
Okay. So I'm going to take a picture of my little bag.
Yeah.
And then I'm going to say that's exactly the picture that I wanted.
Yeah.
And if it's going to tell me the date,
Yeah.
I don't have the location open, but if I open it,
Yeah.
it's going to tell me that I'm actually at BCC.
Right.
And then I can add if it's captive or cultivated.
Okay.
And I can actually have a project. If I add it, so first of all, if I can go and it could tell me what I saw. And it's actually going to come up. This is a lanternfly. So it's like, okay, this is actually a lanternfly. And you can be like, oh, I actually agree with that or I don't agree.
Okay.
So I can already put in something. But then what's great and what's going to happen, let me just go back. I don't want to save this one because it's not real. But for example, you see here,
Yeah.
I took the picture of these and then people went in and actually expert, they do it out of the, as
Yeah.
volunteer,
Yeah.
but they confirm or even add the species that it was. They're like, oh, this is actually an Asian lady beetle.
Yeah.
And if you get like 2 or 3 confirmation, is research grade. This gets into a database that can be searched worldwide.
Okay.
So theoretically,
Very
if I was
nice.
an entomologist, I could be
Yeah.
like, hey, how many ladybugs were
Yeah.
in the Bronx
Yeah.
in 2023 that people actually
Right.
recorded?
Yeah.
So this is a good way for the students to
Very nice.
actually
Yeah.
look at biodiversity.
So
take the
this
time.
app is available
It's available for free
for free. Okay.
in ecology.
Okay.
So if we want to do something that is more of a in-depth project, eventually
Yeah.
we can ask them, because
Yeah.
off the top of my head, I think it was the same thing for you. We are thinking already about activities that we were already kind of doing with the students, maybe individual and not full group and so on, that can be done. But then if these, apart from like sensitizing the students, maybe you also want to collect the data and look at the data and actually do something that is a little more of a
It is not only data for the purpose of our understanding, but ultimately these data will be shared with the government administration, local administration, where it will be helpful then. Because we have to convince them with these data only. Because that there is a gap between the administration and the academic institutions. And academic institutions can play very good role to convince with science that how we need to protect our rivers while we are doing town planning, while we are planning for some treatment plans, what is important, what kind of microbes are playing the role, how at the elemental level, the molecular level, that how they work. These are very, there is a lot of gap while designing of all these things. So it will be a regular monitoring. Whether I am there or not, it doesn't make any difference. As a Fulbright, we start with a process and then it needs to be continued. And it should be a regular kind of internship, you can call it, with your student, where, in every subject. Like, because today in India also, in the new education policy, we are trying to do with how, with community handholding, how every subject can be taught. And in that manner, that whatever the study is being done in the community, that even the government administration, then they are shown, and then what needs to be done in this area now.
Right.
So that way it will be a regular kind of.
Right. So what I was thinking with bringing in Laura into it, maybe, is that because you guys are looking for cures for your Bio 21, right? And there's a
right.
and I think there's a section of that course that has ecology. So like if you can design an experiment, or you can design a project where you can be collecting data that can inform government agencies about the changes that are going on,
So
different
already we have
designs.
prepared a concept note.
Right.
Now that details I'll be sharing with her, how we will be implementing it.
Right. So if you want something,
Maybe for a one visit we do, we show them first. That would be our first survey, we can call it
Right.
at. And on that day, certain seeing the things of all the flora, fauna, and how the water is flowing, and from where what all things are, like exact situation.
right.
Like if at all some the pollution sources are there, what are they? In around we can see what are there.
Right.
So we can show that how catchment areas impact the river. See, students don't know otherwise these
Right.
things. So from that point of view, we have to take to the field.
Right.
So I don't think it should be only ecology student. It can be any student.
No, I'm just saying. I'm
This
not
is
saying that
actually a general
okay.
bio class.
this is a
Okay.
general
intro
bio
class.
class.
Okay,
I'm just saying
okay.
that like if you want something to be,
Yeah.
you want it to be sustainable, right? So
Yeah,
even
right,
after you leave,
right,
You want it to
absolutely.
still be generating. So what I'm saying is
Absolutely.
that if you can
A regular monitoring.
right, so if you can incorporate that
Yeah.
into the cure for that
Yes.
classroom every
Yes.
semester that the
Yeah.
class runs,
Yeah.
you will
You're
be
gonna
generating
collect some
data.
So that is why we have spoken to provost that in the curriculum itself we have to embed these things. Okay, but to begin with, first let us see the response. How students feel when they go to the field and do such kind of a study or
I can
the
already
connected.
tell you because we do similar kind of studies, again, like limited, and it's mostly to expose the students to, how to do research.
Yeah.
So, for example, with Jake last semester, we went out to man-made tide pools on Randall's Island. We collected the water to do DNA metabarcoding of the. We just did 16x for some of them because our barrier is the cost, obviously, of
Right.
metabarcoding still. And then I had some high school students, G and the
Okay.
and the biodiversity. Well, we have all these companies that are making these like man-made tide pools that are
Right.
supposed to support biodiversity, and we're like, okay, so let's see.
So they are creating these kind of
Right, we have a bunch of like
Pools?
this different structure all over New York that
Okay.
are specific companies that again are using material that is actually supported to biodiversity instead of using cement.
okay.
And so I was like, well, why don't we go and see what the biodiversity is in these tide pools?
Yeah.
And so that's what we're
What do you
doing.
call it as tide pools?
They're tide pools. They're literally little. I have pictures because
Huh.
I actually, I think we have the poster of the kids someplace.
Okay.
I don't know. But I can literally show them to you.
I have both as already.
Let's see if it brings up. Like this is not Randall's Island.
Okay.
Let me see if I can get the images. Sorry, this computer is super slow. But these are the kind of like things that they
Okay.
build.
Okay.
So they are.
Just zoom it. Okay. Okay. So
They
what
are
they
so-
call it as?
These one are tide pools, meaning that when the tide goes up and down, they
Okay.
can fill up or they can. And
Where they have built this?
these the
Near to
one.
the Bay areas or where?
No, the one that we studied is actually they're in Randall's Island,
Okay.
but they also have them in other parks in New York.
They're along the Bronx Kill, right?
You know what? The Bronx, they're not along the Bronx Hill. They're actually the one that we studied are along the.
So what is that eco?
This is
Econcrete.
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 Oops, but there are other companies. This is the
Okay.
one that we
Okay.
were,
Okay.
is the one that was making the.
So why
the
they are
stuff.
constructing these?
So the idea is that having all that cement along the river was actually affecting the biodiversity because it's
Okay.
releasing like CO2, is releasing other things that were changing the quality of the water.
So what they are using in this then?
Well, that's proprietary, isn't it? I don't know what they're using.
Because
but-
nowadays a lot of that ash is being used, thermal plants, ash and all. They make bricks out of it and all. Then they call it as more eco bricks and all.
Well, again, there's a lot of like different companies, and I think that whatever they're making is proprietary. But allegedly, these are materials that are actually supporting the biodiversity. So, we have them. But we went out, and we had to be there at 6:00 in the morning because
Yeah.
the Harlem
High
River
tide and all.
is. The river
Yeah.
is coming down, but the tide is
Tide
coming
is
in.
coming.
So you need to be there when it's low,
Yeah,
and you can
yes.
collect the water, filter
Yes.
the water,
Yeah.
and then bring it in, extract the DNA
Yeah, yeah.
and the whole thing, and then
Okay.
we actually were quite surprised because there is quite a diversity within the kind of bacteria that you find in the tide pool. So again, we have limited funds.
So you have identified the bacteria also? You've done
We
the
have some data.
media you have grown or no?
No, we actually did DNA metabarcoding.
Okay.
So we extracted the DNA, we sent it out to be barcoded, which again, it doesn't really get down to the species most of the time, but it gives you an idea of the diversity that you have. And
So you have all the things here in the lab to do it, or you
Yeah.
send it outside? We have.
we, apart from the
You send
metabarcoding, that
the
needs to be sent
sequencing
out.
out. Yeah.
Yeah,
Okay.
the sequencing, which for the metabarcoding is still a little expensive, but
Okay.
I have ways, I have colleagues
Okay.
that
They
if
help
I
in that.
get them at the right time, they may process the stuff for us.
So
it's-
that's what is called as goodwill building.
Well, but then we have
What they are doing, they are not ensuring even that the what bacteria is there and all. They say that we are just trying to comply with the government's norms, that what they want, that is what we are doing, and we feel that that is only correct. Okay, so that's how things are going on, but we need to now convince them with the science. So I need your help for the microbiological study and to identify what kind of bacteria are there and how far we have been able to able to treat them, and the pathogens can be killed, so that before we release the water in the East River, that we are ensuring that the clean water we are releasing. Okay, so because when I went there in the first the second the tertiary tank also, I could see a lot of algal blooms. We need to even identify about the cyanobacteria there. Okay, and I will show you the pictures also. And unless we convince them with these our microbiological studies, I don't think they will be convinced. So, and then ultimate aim is to share these things with the government here, so that whatever the reorientation has to be done with the reinterpretation of the system that we have to do.
Okay, I'm looking. So, what I'm thinking, and I don't know if they're doing metabarcoding, may be expensive. I can't find the file right now. I was not prepared for this because I didn't know. So, what metabarcoding can do, because some stuff you can plate and it's going to grow, but a lot of bacteria have very specific requirements. So
Yes.
they're not going to go. But
Yeah.
the DNA is there.
So, at least from your side, ultimately, what I want, because you have been teaching this, you have been doing research on this, ultimately, when we are doing the analysis part of it, so there you have to give your views and a complete interpretation with the analysis report of the water samples which we will be doing
Okay,
for
yeah, because that
microbial
we were
identification.
not able to do that because
Yeah.
that's not our specialty. So we
Yeah.
were just like collecting the.
Yeah. So now you will be doing more specific and more this thing. So that's what will be our role now. And maybe for the collection also, you can also join me when we have to go for the collection of sample if you have time. That also we will fix according to your availability or whatever the way, because you need to also see the treatment plant.
Right, and that's actually something that will be
Yes.
interesting. So for us,
Yes.
I can tell you something what we used to do.
Yeah.
Because again, we had to deal with the money
Yeah.
that we had. So we got these, and this is actually a it's a manual vacuum pump for for brake fluid.
Okay.
But we were actually using it because we would collect the water and then filter it, and
Okay.
then so we filter the water right away. We would collect the membrane, put it in alcohol, so we would fix anything that was in the water
Okay.
immediately. Whatever you were collecting,
Very good.
we actually had it directly here, and then we would extract the DNA and see
Okay.
what was that.
So this complete procedure, the procedure of doing this, one day if we can do it together,
Okay,
I would also
absolutely.
like to see that, so that we can then come up with a very good report where we can share for why it is needed that our wastewater treatment system should be built based on the specifications. And for that, I will tell you, we are also telling that the treatment plants cannot do much, whereas we have to allow these fecal matters to go to the soil, where soil
I'm also gonna
microbes
tell you
and
something. When you were talking, and I'm thinking about them actually adding something to kill all the bacteria before releasing the water, I'm thinking, well, that's gonna go in the water and it's actually gonna alter the ecosystem
Yes.
of the river.
yes.
So I remember we did again. This is a lot of little things that we had ponds in New York Park,
Okay.
and what they, bacteria population was before and after they sprayed for mosquitoes.
Yeah.
Which obviously they're trying to kill the
Kill
mosquito
yeah,
larvae. But
larvas.
we were like, well, what's the effect on the
It's water body.
on the water body?
Right.
And yeah, we could actually see a difference. That spraying was also affecting some of the.
Yes. You are targeting only the larva, but at a broader scale, if you see, you are polluting again the water body.
Right. So
Yeah.
it's kind of like we know that some things need to be done for the greater good, but we should know what the effect actually
Yeah.
is.
But before that, when we are trying to tell them this is not correct, that is not correct, we have to tell them the solution also. Like suppose if I'm saying don't use this solution for killing the larva of mosquito,
Right.
we will say that go by natural means, release some fishes which eat upon these larvae.
Right,
So-
and that's the thing. I think that
Yeah.
that's the positive part, right?
Yes.
You can't just say you don't
Don't
do
do.
that.
Right,
but
right.
what can we do
What-
that is still
yes,
effective?
yes,
Because again,
yes.
a lot of this stuff is done for the
Yeah.
greater good. It's not done with bad intention.
So that is the reason. See, otherwise to study about this water, you don't call it as treatment plant. You call it as water resource recovery facility. Whereas across the world, we call it as sewage treatment plant. Okay. So with that resource recovery, what has happened, today, the focus is totally on the resource recovery. How we will make gas out of it, how we will make energy out of it. That's what they are thinking about. How this we can put it then in the landfill. But landfill again, you are polluting it with the microbes in that. You have not treated it.
It's
So-
they don't think
yeah.
about some of the things
Yeah.
that are resources as
Yeah,
resources.
yeah.
That's actually
So
what
that's
the
why that
problem is.
Alabama case is still, you know about Alabama.
I do not know
That
about
poop
Alabama.
poop train which was taking all poop from Manhattan to Alabama, and there was a big case, and the court has now said that the poop train corridor also has been stopped. I hear that, but whether it is completely stopped, I don't know. So that still continues. So with this study, I feel with study we should be convincing them that what is required, and we should recommend what
we
will be the
also
right
not
way.
learn what students, what all the students did, right? We went out. So this is in Randall's Island. I'm gonna do it a little bigger. So, this is all the students did all this stuff. So this is Randall's Island. We were interested because the tide pools are actually found in between this big bridge where there's a lot of traffic and a ferry dock. So we went and these are the tide pools. This is when the tide pool is actually
Little
out.
reduced because that is going outside.
Okay, let me see. Oops, wrong direction. Okay.
Yeah.
So this is like at low and high tide, and again, we just wanted them to come out. For the high schoolers, they actually were collecting algae, so they took picture. The PCR needs to be fixed, but for the algae, we were doing DNA barcoding.
So we can even do the algae identification,
Yes.
right? Okay, good. So microbes and algae, I can do
And
too.
then for the metabarcoding, again, what we had, we don't have obviously any control because
Right.
we were thinking actually if we had money to get the water from the river, but this is actually from like different tide pools, and eventually we're doing tide pools and the biofilm.
Good.
And you can get a lot of information. It's not down to the species, but you can see the main group of bacteria, and then you can see what the percentage of them is in different things. And again,